Hey, fellow microbe wrangler. Do you ever get that feeling
What?
like we're in a constant arms race with bacteria?
Oh, yeah, definitely. Constantly trying to keep one step ahead.
Today we're taking a deep dive into this.
Sounds fun.
It is. It's chapter 19 of
Cool.
Antibiotics, Challenges, Mechanisms, Opportunities,
Bye.
Walsh and Wensfitch.
Oh, I know that one. It's a good one.
It is, and it's particularly relevant to you as a medical microbiologist.
Yeah, definitely.
How so?
Well, it's like a roadmap to the future of antibiotics, and you're holding the compass.
That's a good analogy. I like that.
Ah.
So are we talking about like new discoveries or strategies for dealing with resistance?
All of the above. But the big question is this.
Hmm.
Can we actually win this fight? Like, for real? Because
Yeah, I get it.
every time we develop a new antibiotic, it feels like bacteria.
I already have a workaround.
Yeah, that's true.
So what hope do we have? I mean, is there any light at the end of the tunnel?
There is, actually. What's interesting is that researchers are getting really clever about this whole arms race. They're not just throwing darts in the dark anymore.
So what are they doing? Like, what's the game plan?
Well, for one, they're finding ways to revamp those old antibiotics that we thought were
Done. Washed up.
exactly like tetracyclines and quinolones.
Wait, seriously. I thought we'd squeezed every last drop of potential out of those.
I know, right? But it turns out there's still some life in those old dogs. It's all about understanding how bacteria resist those drugs and then finding clever workarounds.
So give me an example. What kind of workarounds are we talking about?
One great example is eravacycline. It's a tetracycline, but it's made in a completely different way, and that's key.
Why is that key?
Because bacteria haven't figured out how to resist it yet. It's like they're looking at it saying, wait, what is this?
So it's not just about changing the molecule. It's about,
Outsmarting them, basically.
yeah, that's amazing.
Yeah.
Are there other examples of this kind of antibiotic makeover?
Definitely. We're also seeing second-generation oxazolidinones like titazolid phosphate and redzolid. They're building on the success of linezolid, which is a great drug, but bacteria started to figure it out.
Of course they did. So are we just delaying the inevitable with these makeovers?
That's a good question. And it leads us to the next big thing.
Which is...
Discovering entirely new antibiotics, like those hidden gems that are out there just waiting to be found.
Ooh, now you're talking.
Oh.
I love the idea of nature's secret stash of antibiotics. What kind of cool stuff are we looking at?
Well, there are these fascinating cyclic peptides and lipopeptides that target lipid 2.
Okay, remind me what lipid 2 is again.
It's a molecule that bacteria need to build their cell wall, so it's pretty essential.
Got it. So these peptides and lipopeptides are basically saying no wall for you.
Exactly, and without a wall, they're vulnerable.
It's like taking down their fortress brick by brick.
Precisely. But wait, there's more.
There's always more. I'm hooked already.
Yeah.
What else?
We've also got myenomycin and its relatives, which go after the enzymes that build peptidoglycan.
Peptidoglycan, that's another crucial component of their cell wall, right?
Right. It's like the mortar that holds those bricks together.
So we're attacking their construction crew from multiple angles. I like the strategy.
And there are even more weapons in this natural arsenal, like arylomycins.
Arylomycins. Tell me more.
They act as signal peptidase inhibitors.
Signal peptidase inhibitors. Okay, now you're getting too technical for me. What does that even mean?
Basically, they mess up the bacteria's ability to process and transport proteins, which are essential for their survival.
So we're not just weakening their walls, we're also messing with their internal operation.
Exactly. We're hitting them from all sides.
I like the sound of that. What other tricks do we have up our sleeves?
Well, we've got gautamines and orodox. They target type 2 secretion systems.
Now, those sound familiar. Remind me what those are.
They're how bacteria secrete toxins and other nasty stuff that make us sick.
Right. So we're basically taking away their weapons before they can even use them.
Precisely. And then there are the heavy hitters, abasomycins and platensomycins.
Okay, those sound like serious contenders. Tell me about them.
Well, abasomycins inhibit an enzyme called PABI synthase.
What does that one do?
It's crucial for bacteria to make folic acid.
Folic acid, isn't that something we need too?
Yes, but bacteria make it differently, which allows us to target that pathway specifically.
So we can hit them where it hurts without harming ourselves. That's smart.
And platensomycins target fatty acid biosynthesis, another essential process for bacteria.
So no folic acid, no fatty acids, no party for the bacteria.
I like it. It's all about exploiting those differences between bacteria and human cells, hitting those critical pathways that are essential for their survival, but not ours.
It's like finding their Achilles heel and going straight for it. That's brilliant.
But here's another question.
Okay.
How do we even find these compounds in the first place?
Yeah, that's a good question. Is it just a matter of randomly testing plants and microbes and hoping for the best?
Not anymore. Researchers are now using smart screens to identify new targets and even combinations of antibiotics that work together synergistically.
Smart screens. Sounds intriguing. What makes them so smart?
Well, traditional screening involved just throwing a bunch of compounds at bacteria and seeing what stuck. It was a very inefficient process.
Yeah, like finding a needle in a haystack.
Exactly. But with smart screens, we can be much more targeted. For example, Merck developed a library of S. aureus strains, each with a single gene silenced.
So they're basically creating a bunch of mutants.
In a way, yes. They then test various compounds against these strains to see which ones are effective when a specific gene is knocked out.
So it's like they're systematically disabling parts of the bacterial machinery and then looking for vulnerabilities.
Precisely. It's a much more strategic approach.
And I'm guessing it's yielded some pretty impressive results.
It has. Remember platensimycin, the compound we talked about earlier that targets fatty acid biosynthesis? That was discovered using this very technique.
Wow, so they basically engineered bacteria to be vulnerable to this new drug.
In a sense, yes, they were able to identify the target by silencing a specific gene.
That's incredible. So these smart screens are like a secret weapon in the fight against antibiotic resistance.
They are. They're giving us a real edge.
What other breakthroughs have they uncovered?
Well, there's kibdelamycin, which is a totally new type of DNA gyrase inhibitor.
DNA gyrase, remind me what that is.
It's an enzyme that bacteria need to replicate their DNA.
So it's essential for their survival.
Exactly. And kibdelamycin binds to a different part of the enzyme than our current drugs, which means it can even work against bacteria that are resistant to those other drugs.
So it's like we're outsmarting them by hitting the same target but from a different angle. That's clever.
It is. And it's a great example of how these new approaches are not just finding new drugs, but also new ways to outmaneuver resistance.
It's like a chess game, trying to anticipate their next move.
Exactly. And there's another example, cystobactamins. They also target DNA gyrase, but with a different mechanism than quinolones.
So we're hitting them with a 1-2 punch.
Precisely. And we're also seeing success with combination therapy, which involves using compounds that boost the activity of existing antibiotics.
Oh, like a tag team of antibiotics.
Exactly. One compound weakens the bacteria, making it more susceptible to the other.
It's like they have no chance. Do you have a good example of this?
A great example is dapdiamide C. It inhibits a key enzyme in cell wall synthesis, and when you combine it with carbapenems, it's incredibly effective against MRSA.
Wow, so it's like a double whammy, hitting them with two different mechanisms of action at the same time?
Exactly. It's a powerful strategy.
It sounds like we're finally starting to think like bacteria, understanding their weaknesses and exploiting them in clever ways.
That's a great way to put it. We're learning to play their game.
But bacteria rely on a lot of different processes to survive. What about those essential processes that they need for energy and building blocks? Can we target those too?
You're on the right track. The chapter also explores targeting bacterial enzymes that use ATP and GTP, which are the energy currencies of the cell.
Okay, so we're talking about messing with their power supply.
Exactly.
I like where this is going. Tell me more.
These enzymes are involved in a huge variety of essential processes, making them prime targets for new antibiotics.
So it's like cutting off their fuel supply.
You got it. And we already have some success stories in this area. For example, quinolones target DNA gyrase, which we discussed earlier, and bedaquiline inhibits the ATP synthase in the bacteria that causes tuberculosis.
So we're seeing that targeting energy production can be a very effective strategy.
It can be, but of course there are challenges.
I bet. Bacteria are always finding ways to outsmart us. What are some of the roadblocks researchers are facing?
Well, one of the biggest challenges is finding inhibitors that are specific to bacteria and don't mess with our own cells after all. We don't want to shut down our own energy production.
That's true. It's like trying to design a weapon that only targets the enemy and not friendly fire.
Exactly. And then there's the ever-present challenge of bacterial resistance.
Right. They're always evolving and adapting.
It's a constant game of cat and mouse.
So are there any glimmers of hope in this area?
There are. The chapter discusses some promising research on pyrazolopyrimidine inhibitors.
Okay, what do those target?
They target Merck, which is an enzyme involved in peptidog glycan biosynthesis in Pseudomonas aeruginosa.
Pseudomonas is a tough one to crack.
It is, but these inhibitors are showing some promise.
So it's like we're finding ways to disrupt their construction projects.
Exactly. But of course, there are challenges.
Like what?
One of the main hurdles has been those pesky efflux pumps.
Efflux pumps, those tiny bouncers that kick out anything they don't like.
That's a good analogy. They can be a real problem for antibiotic developers because they can pump out the drugs before they have a chance to work.
So how do we get around those? Is it a lost cause if a bacterium has efflux pumps?
Not necessarily. Researchers are exploring ways to block those pumps directly or to design antibiotics that can sneak past them.
So it's like a game of stealth and cunning.
It is. It's a constant battle of wits.
Well, I'm glad we have some clever scientists on our side. Are there any other promising targets in this energy production pathway?
There are. The chapter mentions targeting other Myrligeses, which are involved in building the bacterial cell wall.
So we're still focusing on disrupting their construction projects.
Exactly. And we already have a success story in this area with mupirocin.
Mupirocin, now that one sounds familiar.
It's a topical antibiotic that targets ILT-RNA synthetase,
Which is?
another enzyme crucial for bacterial protein synthesis.
Right, so we're seeing that we can disrupt both the construction and the energy supply lines of the bacterial cell.
Precisely. We're hitting them where it hurts.
It's like we're cutting off their resources and sabotaging their building plans all at once.
Exactly. And there's another intriguing case, DTMP kinase inhibitors.
DTMP kinase inhibitors, never heard of those.
They've shown good activity against gram-positive bacteria.
Okay, so they're a potential new weapon in our arsenal.
They are. They target an enzyme that's essential for DNA synthesis, so shutting it down is a pretty effective way to stop bacterial growth.
So we're seeing a diverse range of ATP-dependent enzymes emerging as promising drug targets. That's encouraging. But what about other energy carriers in the cell, like NAD and GTP? Are those potential targets as well?
You're thinking like a true microbiologist. And you're right, those are also potential targets.
So tell me, what's the story with NAD-dependent enzymes? What roles do they play in bacterial cells?
They're involved in a lot of important processes like DNA repair, metabolism, and signaling.
So they're pretty important for bacterial survival.
Absolutely. And the chapter specifically mentions NAD-dependent DNA ligase as a potential target.
DNA ligase, that's involved in repairing breaks in DNA, right?
Right, so if we can inhibit that enzyme, we can essentially cripple the bacteria's ability to repair its DNA.
And that would make them much more vulnerable to damage. So are there any specific compounds that target this enzyme?
There is one that's showing promise. It's a 5-fluoroadenosine analog, and it's effective against Streptococcus pneumonia.
Streptococcus pneumonia, that's a nasty one. So this is a potential new treatment for pneumonia and other infections caused by that bacteria.
It could be. It's still early days, but the research is promising.
This is really exciting stuff. It sounds like targeting NAD-dependent enzymes could be a very fruitful avenue for antibiotic discovery. But what about GTP-dependent enzymes? What kind of potential do they hold?
The world of GTP-dependent enzymes is vast, and it offers a lot of possibilities for antibiotic development. The chapter highlights a few areas that are particularly interesting.
Okay, I'm all ears.
Well, first up, we have some familiar players in protein synthesis, EF2 and EFG.
Those names ring a bell.
They're GTP-dependent enzymes that are essential for bacteria to make proteins.
So disrupting them would essentially shut down protein production.
Exactly. And we actually already have some antibiotics that target them, curamycin, GE2270A, and fusidic acid.
So we're already seeing some success in this area.
We are. But there's always room for improvement, and researchers are exploring new approaches.
Such as?
One exciting new frontier is targeting FTSC.
FTSC, okay, that's a new one. What does it do?
It's a key protein in bacterial cell division.
Cell division, that's how they multiply. So targeting this protein could prevent them from spreading.
Exactly. FTSZ forms a ring-like structure at the site where the cell is going to divide, and it's essential for that process to occur properly.
So if we can disrupt that ring, we can stop them from dividing.
Precisely. And the research in this area is really gaining momentum.
This is fascinating stuff. It sounds like we're on the verge of some major breakthroughs in how we fight bacterial infections. But you mentioned another group of GTP-dependent enzymes that caught my attention, CDI-GMP syntheses and phosphodiesterase. What's the story with those?
Ah, yes. Those are involved in a very fascinating signaling pathway that regulates bacterial behavior, including biofilm formation.
Biofilms. Those bacterial fortresses that are so tough to penetrate with antibiotics.
Yes, exactly. So targeting these enzymes could give us a way to weaken those biofilms.
That would make them much more susceptible to our current antibiotics.
Exactly.
This is really exciting. It's like we're shifting our thinking from brute force attacks to more sophisticated strategies that disrupt bacterial behavior and communication. So like we're learning to speak their language.
It's a paradigm shift in how we approach antibiotic development.
It's opening up a whole new world of possibilities for combating bacterial infections.
Absolutely. It's an exciting time to be in this field.
It really is. But I have to wonder, where will the next generation of antibiotic molecules come from? Will they be synthetic creations or discovered in nature's vast library of compounds?
That's the million-dollar question, and the chapter explores both possibilities.
Okay, let's start with synthetic antibiotics. What are the limitations of our current synthetic libraries?
Well, most of them were designed for eukaryotic targets, not bacterial ones.
So they're not really optimized for finding new antibiotics.
Not really, and as we've discussed, bacteria have their own unique challenges, like efflux pumps and cell walls that are difficult to penetrate.
So what's the solution? Are there any new approaches to synthetic antibiotic discovery?
There are. The chapter introduces this concept called diversity-oriented synthesis, or DOS.
Loss. Okay, what's that all about?
It's specifically designed to create libraries of molecules that are structurally diverse, rigid, and complex, kind of like natural products.
Ah, so it's like taking inspiration from nature's playbook,
Exactly. And it's already yielding some promising results.
such as.
Well, there's gemicin, which disrupts bacterial membranes, and emicin, which inhibits an enzyme called dihydrofolate reductase.
So we're seeing that DOS can produce molecules that have real antibacterial activity.
We are. It's a very promising approach.
But let's not forget about the vast and largely unexplored world of natural products.
Of course not. Nature has been our primary source of antibiotics for decades, and there's still so much more to discover.
But how do we find those hidden gems?
Well, that's where things get really exciting. We're now using cutting-edge tools like genome sequencing and bioinformatics to revolutionize natural product discovery.
So we're not just wandering through the rainforest anymore, hoping to stumble upon a miracle cure.
Nope. We can now delve into the genomes of bacteria and identify potential antibiotic-producing gene clusters.
Even if we can't grow those bacteria in the lab.
Exactly. It's incredible.
So it's like we're unlocking a secret code, reading the blueprints of bacteria to find their hidden weapons.
That's a great analogy. And the potential is enormous.
I bet. There are so many unexplored microbes out there, in the soil, in the oceans, even in our own bodies.
Exactly. And the chapter highlights the potential of the human microbiome as a source of new antibiotics.
Our own microbiome. That's fascinating.
It is. There's a whole ecosystem of bacteria living in and on us, and many of them produce antimicrobial compounds to compete with each other.
So we could potentially harness those natural defenses to fight infections.
Exactly. It's a very exciting area of research.
This has been an incredible journey so far, exploring all these innovative strategies for finding new antibiotics and outsmarting those clever bacteria.
It's a fascinating field, and it's constantly evolving.
But given the challenges of antibiotic resistance, should we be shifting our thinking altogether? Is it time to move away from single-drug therapies and embrace combination therapies as the new standard?
That's a really important question, and the chapter definitely advocates for a shift toward combination therapy.
I can see the logic in that. By hitting bacteria with multiple drugs simultaneously, we're making it much harder for them to develop resistance.
Exactly. It's like overwhelming their defenses with a coordinated attack.
And it also allows us to use lower doses of each drug, which could potentially reduce side effects.
That's right. And it also allows us to use narrower spectrum antibiotics, which are less likely to disrupt the patient's microbiome.
So we're not just improving treatment efficacy, we're also minimizing collateral damage to the patient's own beneficial bacteria.
Exactly. It's a win-win situation.
But I'm curious, are there any practical challenges in implementing combination therapy? It seems like it would be much more complex than simply prescribing a single drug.
You're right, there are challenges. One of the biggest is the need for rapid and accurate diagnostics.
Why is that so important?
Because we need to be able to quickly identify the specific bacteria causing the infection so we can choose the right combination of antibiotics.
It's like a military operation.
Yeah.
You need good intelligence to choose the right weapons.
Exactly. And unfortunately, our current diagnostic tools aren't always up to the task.
So we need faster, more accurate tests.
We do. And the chapter emphasizes the need for developing automated software and accessible databases that can help clinicians make informed treatment decisions.
So it's not just about the technology, it's also about making sure that clinicians have the information they need to use it effectively.
Exactly.
This has been an incredibly enlightening discussion, and I'm feeling much more optimistic about the future of antibiotics. Sounds like we have a lot of promising new strategies and technologies at our disposal.
We do. And it's
Hmm.
an exciting time to be involved in this field.
It really is. But technology alone won't solve this problem. We also need to address those underlying social, economic, and behavioral factors that contribute to antibiotic overuse and misuse. It's not just about developing new antibiotics, it's also about using them responsibly and preserving their effectiveness for future generations.
Couldn't agree more.
Well, after that whirlwind tour of the future of antibiotics, I think we're ready to test your knowledge. It's time for those medical microbiology exam questions. Remember, these are targeted at experts like yourself, so be prepared for a challenge.
Challenge accepted. Let's do it.
All right, here's your first question. What are two examples of new antibiotics that have been developed from existing scaffolds? Take a moment to think about that one.
Okay, two examples of antibiotics developed from existing scaffolds. Okay,
Okay.
I would say tedizolid phosphate, which is a second-generation oxazolidinone, and dalbavancin, which is a second-generation teicoplanin. Both of these drugs represent improvements over their predecessors, and they demonstrate that there's still potential in modifying existing scaffolds.
Excellent answer. You've clearly grasped the concept of building on the success of previous antibiotics. As you mentioned, tetasolid phosphate and dalbavancin are great examples of how we can tweak those existing structures to create new and improved drugs. Ready for question number two?
Hit me with it.
All right, here it is. Describe two smart screen approaches that have been used to identify new antibiotic targets or combinations.
Two smart screen approaches. Okay, I would say Merck's 245 gene knockdown library in S. aureus, and also the screening for compounds that restore beta-lactam sensitivity. The Merck approach, that one led to the discovery of platelsamycin by targeting Fabry, and the beta-lactam sensitivity screen. That one helped to discover dapdiamide C, which can synergize with carbapenems against MRSA. Spot on. You've clearly understood the power of those smart screens in antibiotic discovery. As you explained, they allow us to go beyond random testing and use more targeted approaches to identify promising new targets or drug combinations. They've been instrumental in finding new ways to combat those resistant bacteria. Ready for question three?
Bring it on.
Okay, here's a slightly tougher one. What are the potential advantages and challenges of targeting bacterial ATP and GTP-dependent enzymes for antibiotic development?
Okay, advantages and challenges of targeting ATP and GCP-dependent enzymes. Well, the advantage is that these enzymes are involved in so many essential processes in bacteria, making them really attractive targets for new drugs. But the challenge is finding inhibitors that are specific to bacteria that don't also mess with human cells. We don't want to harm the host while we're trying to kill the bacteria. And of course, there's always the challenge of resistance. Bacteria are incredibly good at evolving and adapting to our drugs, so that's something we have to keep in mind. Fantastic analysis. You've perfectly summarized the delicate balance between the potential and the pitfalls of targeting these essential enzymes. It's that constant game of cat and mouse, trying to stay one step ahead of those crafty bacteria. Ready for the next challenge.
Absolutely. Lay it on me.
All right, question four. What are the key differences between traditional synthetic libraries and diversity-oriented synthesis, DOS, in the context of antibiotic discovery?
Key differences between traditional libraries and DOS. Okay, well, traditional synthetic libraries, those often consist of fairly simple molecules, kind of like what we see in those massive screening efforts. But DOS focuses on creating molecules that are much more complex and diverse, more similar to what we see in nature. And that structural complexity and diversity is important because it allows us to explore a wider range of chemical space, which increases the chances of finding those novel scaffolds that could lead to new antibiotics.
Excellent. You've nailed the key differences. Those traditional synthetic libraries, while vast, often lack the structural complexity and diversity found in natural products, which is where many of our current antibiotics have come from. But DOS bridges that gap. It creates libraries of molecules that are more natural product-like in their architecture, which opens up a whole new world of possibilities for antibiotic discovery. One last question, and this one's a bit more conceptual, but I think you're up for it.
I'm ready. Let's hear it.
Here it is. Explain the concept of combination therapy for bacterial infections and discuss its potential benefits.
Combination therapy. Okay, so the basic idea is that you use two or more antibiotics that have different mechanisms of action, and you use them together to treat an infection. And the potential benefits are pretty significant. First of all, it reduces the likelihood of resistance developing, because it's much harder for bacteria to become resistant to multiple drugs at the same time. And secondly, it can enhance the effectiveness of the treatment by attacking the bacteria from multiple angles. And finally, it can allow us to use lower doses of each individual drug, which can potentially reduce side effects.
Amazing. You've clearly articulated the rationale and the potential advantages of combination therapy.
Huh.
It's a very promising approach that's gaining increasing attention in the fight against antibiotic resistance.
It's exciting to see this shift towards more strategic and multifaceted approaches to treating bacterial infections. It gives me hope that we can stay ahead of those ever-evolving bacteria.
Me too.
Hmm.
Well, you've aced the exam. It's clear that you're not just a medical microbiologist, but a true champion in the fight against antibiotic resistance.
I'm just doing my part. It's a battle that demands our collective efforts. We all have a role to play in ensuring the future of antibiotics.
Beautifully said. And speaking of the future, let's shift gears for a moment and explore a thought-provoking question inspired by the chapter. If you could design the ideal antibiotic, what properties would it have? What challenges would you need to overcome?
Hmm. The ideal antibiotic. That's a great question. Let me think. Well, first and foremost, it would have to be broadly effective against a wide range of bacterial pathogens, including those that are currently resistant to our existing drugs. And it would need to have minimal side effects, so it would be well tolerated by patients. And of course, it would have to be able to evade those resistance mechanisms that bacteria are so good at developing. And ideally, it would be affordable and accessible to everyone who needs it.
That's a tall order, but I love your ambition. It's clear that the ideal antibiotic would be a masterpiece of molecular design and pharmacological ingenuity. But even if we could create such a drug, there are still those broader challenges to consider. Like, how do we ensure that it actually reaches the patients who need it? How do we prevent it from being overused and misused?
You're right, those are crucial questions. We can't just focus on the science. We also need to address the social and economic factors that contribute to antibiotic resistance.
Exactly. It's a complex problem that requires a multifaceted solution.
And it's a problem that we all need to work together to solve.
Well said. This has been a truly enlightening deep dive into the world of antibiotic resistance. We've covered so much ground, from the molecular mechanisms of resistance to the latest innovations in antibiotic discovery. And it's clear that this is a field that's constantly evolving, with new challenges and opportunities emerging all the time. But before we wrap up this part of our deep dive, I thought it would be fitting to reflect on this ongoing battle between bacteria and antibiotics.
I like that idea. It's a constant dance, a push and pull between these microscopic adversaries.
And it's a dance that has profound implications for human health and the future of medicine.
It's a story that deserves to be told.
And what better way to tell it than through the power of poetry? I've actually composed a short poem that captures the essence of this ongoing struggle.
Oh, I'd love to hear it.
All right, here it is. In realms unseen, a silent war, bacteria rise, their defenses soar. But science fights, with knowledge bright, to forge new weapons day and night. From nature's depths to chemist's art, we seek new cures to heal each part.
Wow, that's beautiful. You've captured the intensity and the urgency of this battle. It's a reminder that we can't afford to be complacent.
Exactly. We need to stay vigilant, we need to keep pushing the boundaries of scientific discovery, and we need to work together to ensure that antibiotics remain a viable treatment option for generations to come. This battle is far from over, and it's one that we must win.
You know, it's pretty amazing when you think about it, how much our understanding of bacteria has changed over time. Like, we used to think there were these simple organisms, but
Yeah, simple single-celled organisms, right?
right. But now we know they're so much more complex than that. They have these sophisticated communication networks and survival strategies. It's like a whole new world under the microscope.
It really is. It's kind of like we've discovered they have their own secret society with their own language and rules.
Mm.
It's a bit intimidating, actually, but also,
Exciting.
yeah, exciting. Because it means we can find new ways to fight back, new ways to exploit their weaknesses.
Exactly. This new understanding is driving a revolution in antibiotic development. Instead of just trying to kill them outright, we're now looking for ways to disrupt their behavior, to disarm them.
I like that, disarming them. It's a much more strategic approach.
Yeah.
So what are some of the specific aspects of bacterial behavior that researchers are targeting now?
Well, one area that's getting a lot of attention is quorum sensing.
Quorum sensing. Now that rings a bell. Remind me what that is again?
It's basically a bacterial communication system that allows them to coordinate their activities, like a bacterial social network.
So it's like they're sharing information and strategizing their attack.
Exactly. And if we can disrupt that communication, we can disrupt their ability to mount a coordinated defense.
That's brilliant. It's like intercepting their battle plans. But how do we actually disrupt those bacterial conversations?
There are a couple of approaches. One is to develop molecules that mimic those quorum sensing signals. basically flooding their channels with noise.
So it's like creating a diversion, confusing them so they don't know what to do.
Exactly. It throws a wrench in their plans.
I love it. What's the other approach?
The other approach is to target the enzymes that bacteria use to produce or degrade those signaling molecules.
It's like jamming their communication channel.
Precisely. If they can't send or receive those signals, they can't coordinate their activities.
It's a brilliant example of how understanding bacterial behavior can lead to entirely new therapeutic strategies.
It is. And it's not just about quorum sensing. Researchers are also looking at ways to target other aspects of bacterial behavior, like biofilm formation.
Biofilms, those are tough to crack.
They are. But we're making progress, and researchers are also exploring ways to target bacterial motility.
Motility, so their ability to move around.
Exactly. If we can prevent them from moving, we can prevent them from spreading and causing infection.
It's like trapping them in their own territory.
Precisely. And then there's virulence factor production.
Virulence factors, those are the molecules that actually cause disease.
Right. They're the bacteria's weapons.
So we're not just trying to kill them anymore, we're trying to disarm them.
Exactly. And we're seeing some really exciting results. For example, some researchers have developed compounds that prevent bacteria from attaching to host cells.
So it's like building a wall around our cells, keeping those invaders out.
That's a great way to put it. And we're also developing compounds that block the secretion of those virulence factors.
So it's like silencing their weapons before they can even fire them.
Precisely.
This is truly revolutionary. It's like we're shifting from traditional warfare to a more sophisticated form of defense, targeting the very mechanisms that bacteria use to cause disease.
It's a paradigm shift in how we think about fighting bacterial infections.
And it's giving me a lot of hope for the future.
Me too. It's an exciting time to be in this field.
It really is. But let's shift gears for a moment and talk about another game changer in drug discovery. Artificial intelligence.
Yes. AI is revolutionizing everything these days. And it's having a huge impact on antibiotic discovery, from identifying new drug targets to designing novel compounds.
It's incredible how AI is transforming so many fields. But for those of us who aren't computer scientists, can you break down how AI is actually being used in the hunt for new antibiotics?
Sure. One of the most exciting applications is in target identification. AI algorithms can sift through massive amounts of data, like genomic sequences and protein structures, to pinpoint potential targets that are essential for bacteria, but not for human cells.
So it's like AI is acting as a superpowered detective, finding those bacterial Achilles heels.
Exactly. It can analyze data much faster and more efficiently than humans can, and it can identify patterns that we might miss.
That's amazing. So AI is helping us to find those weak points in the bacterial armor.
It is, and it's not just about finding targets, it's also about designing drugs that can hit those targets effectively.
So AI is also involved in drug design.
Absolutely. We're now using AI to design molecules that fit perfectly into the active sites of those target proteins, effectively blocking their function.
So it's like designing a key that fits perfectly into a lock.
That's a great analogy. AI can help us to design drugs that are much more precise and effective.
This is incredible. So AI is not only helping us to find the enemy's weak points, but also to design the perfect weapons to exploit them.
It is. It's like having a secret weapon in the fight against antibiotic resistance.
But AI's role doesn't stop there, does it?
Nope. It's also being used to predict antibiotic resistance.
Predict resistance. How does that work?
AI algorithms can analyze data on bacterial mutations and resistance patterns to predict which antibiotics are most likely to be effective against a particular strain.
So it's like having a crystal ball that can tell us which drugs are going to work.
In a way, yes. It's not perfect, but it can give us valuable insights that can guide treatment decisions.
This is truly revolutionary. AI is giving us a whole new set of tools to fight back against antibiotic resistance. It's like we're finally starting to catch up to those ever-evolving bacteria.
I agree. It's an exciting time to be in this field.
But even with these powerful new tools, I imagine there are still challenges to overcome. What are some of the roadblocks researchers are facing in using AI for antibiotic discovery?
You're right. AI isn't a magic bullet. One of the biggest challenges is the need for high-quality data. AI algorithms are only as good as the data they're trained on.
So garbage in, garbage out.
Exactly. If the data is incomplete, biased, or inaccurate, the results will be unreliable.
That makes sense. What are some other challenges?
Well, another challenge is the inherent complexity of biological systems. Bacteria are constantly evolving and adapting, and their interactions with antibiotics are incredibly intricate.
So it's not always easy to predict how they're going to respond to a particular drug.
Exactly. And even the most sophisticated AI algorithms can't fully capture that complexity.
It's a reminder that biology is messy and unpredictable. But despite these challenges, it's clear that AI is having a major impact on antibiotic discovery.
Absolutely. We're already seeing some promising results, and I'm confident that AI will play an even bigger role in the future.
It's exciting to think about the possibilities. But even with all these technological advancements, we can't forget about the human element. We need to address those underlying social, economic, and behavioral factors that contribute to antibiotic overuse and misuse. It's not just about developing new drugs, it's about using them responsibly.
You're absolutely right. We need a multi-pronged approach that combines scientific innovation with public education and policy changes.
So it's a collective effort that requires everyone to do their part.
Exactly. We all have a stake in preserving the effectiveness of antibiotics.
Well said. This has been an incredible discussion, exploring the multifaceted challenge of antibiotic resistance and the innovative approaches being developed to combat it. It's clear that this is a global issue that requires a coordinated and multidisciplinary response. We need scientists, policymakers, healthcare providers, and the public all working together to ensure that antibiotics remain a viable treatment option for generations to come.
I couldn't agree more. The fight against antibiotic resistance is a marathon, not a sprint, and we need to leverage every tool at our disposal to win this race. But as we continue this journey, it's also important to ask ourselves some fundamental questions about our relationship with bacteria.
I love that. It's not just about finding new weapons, it's about rethinking the battle itself. So what are some of those fundamental questions we should be asking?
Well, one question is, what if we could completely reimagine our relationship with bacteria? What if we could move beyond this adversarial mindset of us versus them
Right.
and recognize that bacteria are not just our enemies, but also essential partners in our health and well-being?
I love that perspective. It's a reminder that the vast majority of bacteria are actually beneficial or harmless. We rely on them for so many things.
Exactly. Our microbiome, the trillions of bacteria that live in and on our bodies, plays a crucial role in our health. So instead of waging war on all bacteria, maybe we need to find ways to coexist more harmoniously.
That's a beautiful vision. Living in harmony with bacteria. It sounds like a utopian dream.
Maybe, but I think it's a dream worth striving for.
I agree, but how do we make that dream a reality? How do we shift from a mindset of eradication to one of balance and coexistence?
It starts with a deeper understanding of those complex interactions between bacteria and their hosts. We need to move beyond the simplistic view of bacteria as pathogens and recognize their crucial role in our health and well-being.
So it's about seeing them as partners, not just enemies.
Exactly. And that understanding will help us to develop more targeted and sustainable strategies for managing bacterial infections.
It's about finding that balance, that sweet spot, where we can control the harmful bacteria without wiping out the beneficial ones.
Precisely. And it's not just about developing new antibiotics. We also need to explore alternative approaches, like phage therapy.
Phage therapy, that's using viruses to kill bacteria, right?
Exactly. Phages are viruses that infect and kill bacteria, and they've been used therapeutically for decades.
It's amazing how nature often provides the solutions to our problems.
It is, and with the rise of antibiotic resistance, there's renewed interest in phage therapy as a potential alternative or complement to traditional antibiotics.
So it's like we're enlisting the help of viruses in our fight against bacteria.
In a way, yes, it's a fascinating approach.
It is. But what about our own defenses? Can we harness the power of our immune system to fight bacterial infections more effectively?
Absolutely. Our immune system is our first line of defense against infection, and there's growing interest in using immunotherapy to boost our immune response to bacteria.
So it's like giving our immune system a boost, helping it to fight those infections more effectively.
Exactly. And there are a number of ways to do this. One approach is to develop vaccines that target specific bacterial pathogens.
Vaccines, that's a classic approach.
It is, and it can be very effective. But we're also looking at ways to stimulate the immune system to attack bacteria directly.
So it's like training our immune system to be more aggressive.
In a way, yes. And one promising approach is to use antibodies to target bacteria or to neutralize their toxins.
Antibodies, those are like guided missiles that can seek out and destroy specific targets.
That's a great analogy. And we're also looking at ways to stimulate other parts of the immune system, like T cells, to attack bacteria.
This is incredible. It's like we're unlocking the full potential of our immune system.
It is. And it's opening up a whole new world of possibilities for treating bacterial infections.
This has been an incredible exploration of the cutting-edge strategies being used to combat antibiotic resistance. It's clear that the future of antibiotics is far from bleak. There's so much to be hopeful about.
I agree. We're witnessing a renaissance in antibiotic discovery, driven by a deeper understanding of bacterial biology and a commitment to finding solutions to this global challenge.
This has been a fascinating journey so far, exploring the multifaceted world of antibiotic resistance. We've delved into the molecular mechanisms that bacteria use to evade our drugs, the innovative strategies being developed to combat those mechanisms, and the broader social, economic, and behavioral factors that contribute to this global challenge.
It's been an enlightening conversation, and it's clear that the fight against antibiotic resistance is not just a scientific endeavor, it's a societal one. We all have a role to play in preserving the effectiveness of these life-saving drugs.
Well said.